After going this module, you will be able to know about the nature and morphology of hair. You will be able to know the forensic importance of hair. Additionally, this module will let you know the identification and comparison of hair. Follicles are present in the skin or dermis from which protein filaments grow. This protein filaments is known as hair. Human body is covered with skin and hair follicles within the skin. These follicles produced both thick and fine hair depending upon the location of the hair on the body. Hair is composed of the protein called keratin and it is an important biomaterial. Hair is one of the common and important physical evidence encountered in a crime scene. Individualization of human hair, that is, whether it is from head or body, the forceful removal of the hair may have blood or skin with the root. In such cases, DNA typing can be done. There have been advancements in DNA typing technology. Earlier, only hair strand with the root attached could be used for typing, but now with the advanced technology, hair without root can also be used if there are enough number of hair strands. In absence of root, blood or any material through which the typing can be done, appearance, that is the structure and color of the hair, becomes the most characteristic forensic feature of hair. Proper collection and examination of hair can provide the strong corroborative evidence. Differentiation between the cut and forcibly removed or fallen hair can be made very easily. Next, I will be talking about the nature of hair. Hair grows on most of the limbs of the human beings and they also grow on animals. Mammalian species have two types of hairs, long and stiff hair and fine fuzzy and woolly hair. Hair grows from the hair centers called follicles in the body. They are in the shape of fine rods. They may or they may not have fine tips. Some hairs which have fine tips, they acquire almost uniform diameter later on, whereas others do not. Hairs grows to the certain length and they fall off if they are not cut. New hair may come up in their place. The growth rate of hair from other parts of the body apart from the scalp and beard is less. Chemically, hair are made up of proteins, mainly keratin. The hair pigments consist of melanin proteins, uric acids, vitamins, cholesterols and other organic materials. Elements such as lead, iron, arsenic and silicon are also found in the hair. Their elements form the basis for the neutron activation of hair. Slow lead or arsenic poisoning is detected through the excess of poisonous metals in the hair. Due to external contamination, the hair may be found covered with oily substance, that is dirt or dust. Morphologically, hair consists of root, shaft and tip, which have characteristic shape and structures. Pigment distribution and presence of air bubbles in the hair, they give the additional points for characterization. The structure of hair is characteristic in another way. It has three parts: the central medulla, the middle part surrounding the medulla is cortex and the outer part is cuticle. The nature of medulla and its diameter with reference to the total diameter of hair are highly characteristic and often indicate the species of the source of hairs. Further, the structure of cuticle is like the scales of fish and are also characteristic of origin. Physical properties of hair like refractive index, birefringence and density also differ not only from one species to another, but it also differs from one individual to another individual. The hair undergoes changes. These changes in the same individual depend on the food, habits, climatic conditions and age. Besides the external treatment, bleaching, dyeing, cutting and combing have definite effects on the hair. Now, you will be knowing about the forensic issues that are associated with hairs. Mutual transfer of hair due to contact is a classic example of Locard's principle of mutual exchange of traces. Hair, if properly searched, are found in large number of cases. It is one of the earliest type of physical evidence which was considered, collected, evaluated and utilized in forensic work. Individualization of hair with regard to its source has been eluding the scientist up till recently. The identification of hair is not conclusive at the present stage of development except through DNA typing. If root hair are present or through mitochondrial protein analysis. In extremely rare cases, presence of some individual dye, contamination, some extraordinary defect or disease in the hair, however, may permit individualization of the source, or on the other hand, it is possible to exclude some persons as the possible sources of questioned hair. This exclusion, the possible corroboration and its identification now with new techniques make the hair evidence one of the important pieces of physical evidence. The evidence establishes the possible presence of culprit at the scene of occurrence. It indicates the contact if transfer of hairs has been taken place. It also indicates the route of ingress or egress if some hairs are retained, a point of entrance or exit. It helps to identify the vehicle which was used in the commission of crime. It may also establish the ownership of the article left at the crime scene. The hair evidence is often not easily seen, hence it goes unnoticed and it does not bother the culprit. Yet, if they carry blood or blood cells through DNA typing, they permit the definite identification of culprit. Hair evaluation have also been utilized to determine the nature, quantity and the period over which the poison had been administered in slow poisoning cases. In case of Napoleon, the nature and quality was determined after about 140 years. The hair evidence as corroborative evidence is available in most of the serious cases, especially in offences against person. Every human being shed hairs, normally about 10 hairs a day. The falling rate if person is under emotional stress such as in rape or murder attempts in case increases, the hair are therefore likely to fall at the scene. They can form important corroborative link if collected properly. The hair are ordinarily available in murder, assault, road accidents, rapes, abortion, bestiality, housebreaking, maiming, poaching, worrying and thefts of fur of animals, and identification of hair such is not an easy task. Extensive experience is required in this field as there are variation in the hairs of same person at some as well as at different times. Further, there are similarities in the hair of the some persons. However, recent advances, especially in the DNA typing of the hair root cells and mitochondrial proteins, it hold promise for definite acceptable conditions whenever the body cells are available. Next, I will be talking on the morphology of hair. Hair is an appendage of skin that grows from the hair follicle. It has root, a shaft and a tip. The portion of the hair that lies in the follicle is known as root. Root is surrounded by the loose connective tissue known as root sheath. It lies in the dermis. The shaft grows and projects out of the skin. Distal end of the shaft is known as the tip. Vacuoles or air bubbles are present in the hairs. The hair is generally consists of 3 zones, which includes cuticle, cortex and medulla. Examination of these 3 zones is done for identification and comparison of hair. Simple microscope can be used to observe hair and its characteristics. First, I will be talking about the cuticle. Cuticle is the outer layer of the hair. Hair is resistant to chemical decomposition and can retain its structural characteristic over a long period of time. These 2 characteristics makes hair a good evidence to establish individual identity. The cuticle and the outer covering of hair is mainly responsible for this resistance and stability. Scales are present on the cuticle. These scales always point toward the tip of the hair and overlap each other. Hardening and flattening of special cells form the scale. These can be of various types such as serrate, imbricate, flattened, crenate and elongated. For example, human hair has imbricate cuticle. These cells get hardened while progressing from follicle. The best way to observe the cuticle is molding. Transparent fingernail polish or soft vinyl is used to place a layer on a slide. After placing a layer, hair is laid on the top of it. The hair is removed after drying of polish. A mold is created which can be observed under microscope. Another way to examine cuticle is by using electron scanning microscope. Maintenance and cost of such instruments is high. Animal scales appears as shingles on a roof. For species identification, various scale patterns formed on animal hairs are used. Next part is the cortex. Cortex is present within the cuticle. It is like a hollow cylinder composed of fine fibers if protein material. Spindle-shaped cortical cells aligned in a regular pattern forms the cortex, which are present parallel to the length of the hair. It comprises the bulk of hair. It contains color imparting pigment granules, which makes it important for forensic examination. Shape, color and distribution of these granules, they are compared among different hair samples. Hair is placed with few drops of water on a slide and then it is covered with a cover slip. It is then observed under microscope to observe the feature of cortex. The next part of the hair is medulla. A collection of the cells which runs through the center of the hair and gives canal-like appearance is medulla. Medulla is one of the predominating feature in the animals. They can occupy even more than half of the hair's diameter. Estimation of the width of medulla in hair, it is done by calculating the medullary index. It is the ratio of the diameter of medulla to the diameter of shaft and is represented by capital I. For example, medullary index of human hair is 1/3. It is not necessary that all human hair will contain medulla. It can be absent. The appearance of medulla slightly varies in a single individual's hair if it is present. It can be classified into various types, that is simple, compound, globular, fragmented, continuous, interrupted or absent. For example, head hair of human lack medulla, or if present then it is fragmented medulla. In humans, Mongoloid race is an exception. They do not have fragmented medulla, they have continuous medulla. It is present in most animals and is either continuous or interrupted. Medullary characteristics are identical among species, but it may vary within the same animal. Identification of species is done by general shape of medulla. The cylindrical medulla is present in many animals as well as humans. The pattern shaped medulla is present in other animals. For example, deer hair appears as oddly shaped spheres crowded close together. Now, I will be telling you the difference between the human and animal hair. The identification of origin of hair is very important in forensic investigation. There are several differences in human and animal hair. To confirm whether the hair sample found at the crime scene belongs to the animal or human, a number of characteristics are observed and comparison is made based on the results obtained. For example, the texture of the hair in case of the humans, it is fine and thin, whereas in case of animal the texture is coarse and thick. In case of human hair, the scales of the cuticle are small, flattened and surround the shaft completely, whereas in case of animals, the scales are large, polyhedral, wavy and do not surround the shaft. Medulla, in case of humans, it is narrow, it may be absent, fragmented or discontinuous. In case of animals, the medulla is broad, it is always present and continuous. The next characteristics on the basis of which the animal and human hair can be differentiated is cortex. Cortex is thick in case of humans, it is 4 to 10 times broad as medulla, whereas in case of animals it is thin and rarely more than twice the breadth of the medulla. The medullary index in case of humans it is less than 0.3, whereas in case of animals it is more than 0.5. Pigments in case of human hair, they are more towards the periphery of cortex, whereas in case of animals they are uniform, peripheral or central, whereas the precipitin test with intact root is specific for both humans and animals. Now, I will be talking on the identification and comparison of hair. The examination of the hair evidence found at the crime scene is the first step which should be performed. Hair should be examined to establish the origin of the hair, that is, whether it is human hair or animal hair. Comparison of hair with victims and suspects should be done in case of human hair. Differentiation of human and animal hair can be done with ease. Human hair comparison should be done with extreme caution. Variable characteristics can be exhibited by hair not only from one individual to another but also within a single person. The morphological features of hair, they can be observed by careful microscopic examination. These features help in differentiating between human hair and animal hair. The control sample should always be considered before reaching a conclusion. The main characteristics that should be examined include the medullary index, scale structure and medullary shape. As forensic evidence, hair is used to compare and determine whether the hair recovered at the crime is of same origin as that of suspect or not. Scalp or pubic hair are most commonly found at the crime scene. Color, length and diameter of the hair are the most important characteristics that should be matched while comparing hair samples. The presence or absence of medulla, its distribution, shape and color intensity of the pigment granules found in the cortex are also important characteristics for comparison of hair. Hair grows at an average of 1 cm per month. A number of features are observed and compared for identification through hair, which include the color of hair, area of origin, presence of micro residual material, unusual appearance of hair and pigments. First, we will talk about the color of the hair. The general color of human hair varies from the black, blackish brown or reddish brown. In absence of pigmentation, they turn grey and then eventually white. Dyed or bleached hair can be predicted by examining under microscope. Time passed since coloring can also be estimated if growth of the hair since hair was colored or bleached is present. The cortex is colored throughout when dyed or colored. Yellowish tint is observed on bleaching hair due to removal of pigments from hairs. Granules are present in naturally colored hair. The texture of naturally colored hair is similar to a picture colored by a crayon, whereas smooth uniform color is displayed in case of artificially dyes or colored hair. The next is area of origin. The hair of one part of the body differ from the other part of the body. Scalp hair are usually longer than hair on any other part of the body. The scalp hairs usually have flat tips as they are most often cut. They are soft with tapering ends. Constant combing causes the tapering end to fray out in a brush like form. Periodical cropping shows sharp cut tips which becomes blunt and round about a week later. On cross section, the hair appear oval or circular in outline. Pubic hair are curly, short and stout. Beard and mustache hair are coarse. The tips are mostly cut. On cross section, they are oval and more flattened as compared to the scalp hair. Hair from the nose, eyebrows, eyelids or ear are short and stubby with wide medulla. They taper rapidly to form a fine tip. The next is presence of micro residual material. Sometimes due to external circumstances, the residual material can be found on hair. Materials like blood, grease, paint, powder, soil and dust, it can be found on the hair which can further lead to important clues when examined. The next is unusual appearance of hair. Due to clipping, pulling, burning or crushing of the hair, an unusual change in the structure of hair is observed. Normally, the tip of the hair is pointed, but when it is cut, it becomes flat. Due to pulling the diameter of shaft, it reduces the length of the hair. If the hair is pulled beyond its threshold limit of the elasticity, then it breaks and the end becomes cone shaped. The next is pigments. Various pigments are present in hair. One of the pigment is melanin, which is responsible for the color of the hair. Presence of large amount of eumelanin result in dark black hair. In case of red hair, pigment called phenomelanin is present. According to the type of pigments present and its distribution, the comparison between two specimens can be done. The pigment distribution and internal structure can be helpful in the identification between species. Students, next I will be telling you the identification of race on the basis of hair characteristic. Through the gross characteristic of hair, a person's identification race can be predicted. A cross section of it can show the structure, diameter, the thickness of cuticle, pigments, hardness of hair, which can be helpful in identification. For example, in Negroid race, the pigmentation is more and their distribution is also disturbed. Hairs are hard, small, curly and stiff in nature. In Mongoloid race, hairs are usually straight and have more pigments. Their cuticle is thick and cross section is circular in shape. So students, let us see what we have studied in this module. Hair is an appendage of skin that grows from the hair follicle. It has a root, a shaft and a tip. Hair pigments consist of melanin proteins, uric acid, vitamins, cholesterol and other organic materials. Elements such as lead, iron, arsenic and silicon, they are also found in the hair. It is one of the earliest type of physical evidence which was considered, collected, evaluated and utilized in forensic work. The evidence establishes the possible presence of culprit at the scene of occurrence. Hair evaluations have also been utilized to determine the nature, quantity and the period over which the poison had been administered in slow poisoning cases. The hair are ordinarily available in murder, assault, road accident, rapes, abortion, bestiality, housebreaking, maiming, poaching, worrying and thefts of fur and animals. The structure of hair is characteristic in another way. It has three parts: the central medulla, the middle part surrounding the medulla is cortex and the outer part is cuticle. Hair should be examined to establish the origin of the hair, that is, whether it is human hair or animal hair. Variable characteristic can be exhibited by hair not only from one individual to another but also within a single person. A number of features are observed and compared for identification through hair, which include color of the hair, area of origin, presence of micro residual material, unusual appearance of hair and pigments. A cross section of hair can show the structure, diameter, thickness of cuticle, pigmentation, hardness of hair, which can be helpful in the identification of race.